Hello everyone. In today's lecture, we shall talk about the translocation of organic solutes. So let's define what is translocation of solutes. The movement of organic food materials or solutes in a soluble form from one place to another in higher plants is called as translocation of solutes. It is called solutes because the food materials move in a dissolved state. Translocation of solutes is very important in plants because 1. The green plants manufacture their own food and it must be supplied to various parts of the plant body where it is required for their consumption as well as storage. Also, during the germination of seeds, the insoluble food material of the seed is converted into soluble form and is supplied to the growing regions of young seedling till it has developed its own photosynthetic system, that is, leaves. Translocation of organic solutes always takes place from a region of its higher concentration, that is, supply end, to the region of the lower concentration, that is, the consumption end, what we call as sink. In other words, we can also call it as a source sink concept. Translocation of solutes may take place in different directions. It may take place in a downward manner, in an upward manner, or it may take place laterally. In downward translocation of solutes, the organic material is manufactured by the leaves and translocated downward to the stem and roots for consumption and storage. While in upward translocation, it mainly takes place during the germination of seeds or tubers, etc. When stored food, after being converted into soluble form, is supplied to the upper growing part of the young seedling till it has developed green leaves. Upward translocation of solutes also takes place from the stem to the young leaves, buds, or flowers which are situated at the tip of the branch. While lateral translocation requires the lateral movement of organic solutes from the cells of the pith to the cortex. To explain that translocation of solutes takes place through the phloem, a simple experiment can be demonstrated called as the ringing or girdling experiment. A potted plant is taken, a part of the bark or the stem along with the phloem tissue is removed, keeping the upper region as foliated, and we can defoliate the lower part of the stem. After some time, we see that the upper foliated part slightly swells up at its lower end, while the lower part of the stem, which is defoliated, dries up. The reason because the food gets accumulated, the food that is prepared in the leaves gets accumulated at the lower part of the foliated region and is unable to move to the lower part of the stem because of the absence of the phloem tissue. Thus, this experiment is a clear demonstration. Transport of radioactively labeled substances through phloem has been demonstrated using micro-autoradiography. When a leaf of the plant is allowed to photosynthesize in presence of labeled carbon dioxide, and the stem is then excised and processed for micro-autoradiography, it has been observed that the translocation of carbohydrates labeled with carbon isotopes takes place through the stem. Most of the isotope is confined to the transport pathway. High densities of C14 labeled sugars are found in the sieve elements, demonstrating that these cells constitute the transport pathway. In this picture, you can easily see the diagram of the structure of phloem. The phloem is a vascular tissue. It is a food conducting tissue which comprises of 4 elements, namely sieve tubes, companion cells, phloem parenchyma, and phloem sclerenchyma. The main tissue that is involved in the movement of food is the sieve tubes. To explain the mechanism of translocation through phloem, it was well demonstrated by the pressure flow hypothesis, or the Munch mass flow hypothesis, proposed by a German scientist by named Munch in the year 1930. According to Munch, the translocation of organic solutes takes place en masse through phloem along a turgor pressure gradient, that is, from a region of higher turgor pressure to a region of lower turgor pressure, that is, from a region of supply end to the region of the consumption end. The principle involved in this hypothesis can be explained by a simple physical system. To demonstrate the pressure flow hypothesis, Munch created a physical system comprising of two glass osmometers A and B connected through a tube T. In the osmometer A, concentrated solution was filled in, while a dilute solution was filled in osmometer B. Both osmometers were then placed in a beaker containing water. Due to higher concentration of sugar solution in osmometer A, water enters into osmometer A because of the turgor pressure gradient, and as a result, because of the increase in the turgor pressure, there is mass flow of sugar solution from A to chamber B through the tube T till the concentration of sugar solution in both the osmometers becomes equal. But if there is a continuous supply at the end, that is osmometer A, and its removal at B continuously, then the equilibrium will never be reached. So according to Munch, such a system exists in plants also, where chamber A corresponds to the mesophyll cells of the leaf, where food is being constantly produced, and chamber B, or the osmometer B, corresponds to the consumption end, that is, where the food is being constantly consumed or utilized for various metabolic activities, or it is utilized for the growth and development of the plant. Let us look at the demerits of Munch's hypothesis. This hypothesis accounts for the translocation in only one direction at a time. That means Munch's hypothesis only proved a unidirectional movement of solutes and not bidirectional as seen in plants. There is a considerable doubt regarding the magnitude of the turgor pressure at the supply end, which may not be sufficient enough to overcome the resistance offered by the sieve plates in the translocation of solutes through sieve tubes. The turgor pressure may not always be higher at the supply end. This hypothesis is based on purely physical assumptions and does not take into account the fact that whole of the translocation process is dependent upon the plant's metabolism and the metabolic energy. Let us have a look at what is vein loading and unloading. So substances are, or the organic food material is prepared in the mesophyll cells of the leaves. Now these substances that are prepared have to migrate from one cell to another cell through many barriers, which is called as the transcellular transport. So these sugars produced in the photosynthetic cells have to move through several parenchyma cells before they reach the seed elements of the vein in the leaf. This movement of sugars or photosynthates from mesophyll cells to sieve tube elements in the leaf is called as phloem loading. The transfer of sugars or photosynthates from sieve tube elements to the receiver cells of consumption end, that is sink, is called as phloem unloading. In this image, you can clearly see what happens to the photosynthase that is produced in the mesophyll cells. In the step 1, you can see water is absorbed from the neighboring cells, which increases the hydrostatic pressure in the mesophyll cells. In step 2, the phloem loading takes place where the transfer of organic food from mesophyll cells to the sieve elements or the sieve tubes. Step 3, water from the neighboring xylem enters into the sieve elements by osmosis. In step 4, we can see the hydrostatic pressure difference that drives the solute molecules from sieve tubes to the sink region. In step 5, we can see what is called as phloem unloading, where the food is totally translocated from the sieve tube to the other parts of the plant where it is required. The phloem loading mechanism occurs in two ways. One, through the apoplast, that is, through the cell walls. They are loaded into the companion cells and then into the sieve element, or through the symplast, that is, through the plasmodesmatal connections. Here we can study a model for the sucrose proton co-transport. So how sugars are transported across the plasma membrane of the sieve element. So during this process of Wayne loading, enzyme ATPase hydrolyzes a molecule of ATP, which pumps two protons out of the cell. Thus, outside of the membrane of the sieve element becomes more positive than inside. On the outside of the membrane is a sucrose proton translocator. So when the translocator is negatively charged, it accepts a molecule of sucrose and proton. This complex migrates across the membrane mainly as a result of the electronic or electrical imbalance. Sucrose and protons are then released into the interior of the sieve element. The sucrose proton translocator is called as a symport which helps in the movement of linked entities in the same direction. Other types found in higher plants are the uniport, which helps in the movement of single entities across the membrane at one time, while antiport helps in the linked migration of two entities in opposite direction at the same time. Thank you.